Okay, we will now have the Q&A session. You can approach the microphones if you would like to ask questions. So, I need to put this on. Thanks. Okay, so this was interesting. It's nice to see how our fields of study are so different, but somehow we all end up in the same session about life sciences and biomedicine. And we already have some questions. Yeah, you're welcome.
Hello, I have a question for Emma. So if I understand correctly, you use the microfluidic chip to model like the vasculature fluid dynamics, is that correct?
Approximate, I'd say. Like, it's not a perfect mimicry.
Yeah. So could you comment on like the limitations of this model and what you can and cannot infer from the information you get?
I think our first initial goal is just to see if we can see any differences across different flow rates. And the idea is that we'll have 4 channels side by side and we'll change the flow rates in each channel. So it will be very comparable between the different flow rates. The biological variation will be controlled for. And so we'd see over a range of flow rates which would be close to what we'd see in the vasculature, whether that affects the invasion process, because no one's ever looked at this before. But we're not going to be close to what is actually happening in the body. The hematocrit, the density of the blood is going to be different. We're not going to have like the epithelial lining that you would have in a blood vessel. So I think this is the starting point, and actually to fully mimic what happens in the body, there's a long way to go, if that makes sense.
Sure, cool, thanks.
Yes, please.
Hello, I have a question to you, Itai. I would like to ask you, have you tested whether your mutants, which cannot undergo the phase separation in the complex with the RNA, still bind the RNA or not?
Basically, the interactions with the RNA are not constant. Like it keeps interacting and releasing, interacting and releasing. So that's what makes these liquid condensates. They're always in a flow of movement. They're not really binding, like sticking to each other. And when they're sticking, like it starts, when you have some interactions, it stays in this liquid phase separated form. And if the interactions are stronger, it goes to some kind of gels. And if it's even stronger, it goes to aggregates, fibers, and all that. So it might, sometimes it can bind and then just remain soluble. But I have not tested whether it's something, when we have no phase separation, we don't really not yet know what happens, and it's quite interesting for the community also, actually. Yeah.
Thank you.
Thanks. Yes, please.
Okay. Hi, my name is Phyllis, and the question is for Emma as well. Thank you for your presentation. I'm just curious about the first part, which is the invasion of the merozoites into the red blood cells. How would that be translated into, like, say, an anti-malarial drug? And what about the stages preceding that, like from the liver, then what if we prevent the blood cells from, the merozoites from invading red blood cells?
Yeah. I think the window of invasion that I'm looking at is one of the key points we have for particularly vaccines to work, because for most of the rest of the life cycle, at least in the blood stage, the parasites are within the human cell. So an antibody isn't going to really be able to block part of the malaria's life cycle. So if we can have a vaccine against one of these proteins that can produce a high enough antibody titer, and we can block invasion, then we can help to reduce the symptoms or even kind of prevent malaria in patients, if that makes sense.
So it's mostly looking at the stage where you get the febrile illness and not necessarily when it actually invades?
Yeah, so I think there are some vaccines that have gone into clinical trial and they're promising, but they're only about 70% effective. And I think that's because it's incredibly hard for the immune system to actually block the malaria. So I think it's thought in the field that to have an effective treatment, we're going to have to target lots of different points of the life cycle to really reduce the burden of malaria in people to the point at which you don't get symptoms and transmission. And I think this won't be the key to curing it, targeting this window, but it could be part of a treatment that also targets the liver stage and maybe even transmission.
Great, thank you.
Thank you.
Yes, please.
I have two questions, if may ask. So the first one is for Itai. So for a proper drug delivery system, you should be able to use this system in like physiological salt conditions. And I just noticed that your tests are quite low. It's 14 millimolar salt, and usually it's 150.
Yes.
So I wonder if at higher salt you have some issues in terms of encapsulation rates.
Yes, we do have issues, but the thing is with living systems, and I'm already doing like tests with cell cultures. So this is a proof of concept that you can use this UV trigger to create release. So now to get it to work in real life, we need to. We already did design new peptides and new systems with other materials that can remain stable within the cells and also not be too much toxic to the cells. So we are working on the redesign of the system to make it available for drug delivery for real. We're not going to go all the way, of course, because
Yeah, it's not.
but we'll get there.
And the other question is for Siriphong.
Yeah.
So if I understood correctly, the mechanism that you propose is based on computational analysis, or like the intermediate.
Firstly, I try to study from the DFT calculation first, and also from the literature, because in our group also study about the Grignard reaction by using the copper catalyst, and also, but we don't know which step is rate determining step, and also the computational data suggest the step that transfer the carbon nucleophile to the C4 of the pyridine is rate determining step, right? And also we use the 13 carbon KE to solve for that step.
Yeah, because I imagine it's quite unstable, and I was wondering if you can trap it using different derivative
No, for the
and analyzing it, I don't know, NMR or another technique.
Oh yeah, and for the 13 carbon KE at natural abundance, right? We determine the number of the 13 carbon on the sample, and also in this reaction we didn't isolate the intermediate. Yeah, we completely to isolate the product or the remaining starting material. Yeah, and then we put in the NMR to determine the quantitative 13 carbon NMR. Yeah.
Okay, thank you.
Yeah, but if you want to know the experiment, you can discuss with me later. Yeah.
Thank you. So while you're thinking if you have any more questions, I want to ask you something, Siriphong, if I may. I'm not very strong in organic chemistry, so I was wondering, like, what kind of at the end point, what your methodologies, what kind of molecules they could impact in efficiency of synthesis?
Yeah, but now we just to develop the method to make the interest molecule. It's like the cross structure for the medicine, or yeah, it's probably this methodology is able to reduce the step for synthesis of medicine, something like that.
Yeah, so it could create more efficient synthesis for biomedicinal.
Yeah, but I think in the future it's probably is useful.
Yeah, but it's still like far away from implementing in the industry or something like that right now.
Yeah, but industrial scale is probably we can work on this because we try to make the gram scale synthesis is also efficient, right? Because the small scale and gram scale, the result is quite similar. But I think if we can develop to the industrial scale, is possible. Yeah.
Okay, that's nice. Okay. Yes, please.
I have a question for Siriphong. I am wondering, like, what was your criteria when choosing the Lewis acid to be used in your reaction? Because I noticed that different Lewis acid lead to a completely different result.
Yeah, but I think it's depend on the Lewis acid, right? Because when I use the silicon base, it cannot produce the enantiomer selectivity. But when I use the boron base, we got the best result. So I think because of the binding of the silicon with the nitrogen is more stronger, so it make the energy barrier between the transition state pro-S and transition state pro-I is quite similar, so it's kill the enantioroselectivity of the product.
Okay, thank you so much. Yeah.
Thank you. So we still have 5 more minutes. And Emma, I want to ask you something regarding the first question that was asked. So basically, what is the medium that you use inside the microfluidics?
It's a cell culture medium that mimics the plasma of blood. So it has a similar protein concentration to the blood, and it's what we know the parasites are happy dividing in. But I guess ideally we'd switch to using closer to real blood in the future.
Yeah, I understand that you want to get to the endpoint of in as much close as living human physiology, but I might have missed that, but you used several flow rates for the...
That will be the next step, yeah. So the different channels will have different flow rates.
And what about changing flows, like in the blood, like the flow rate changes during the experiment?
Yeah, it
Do you
goes...
plan to do that?
Yeah, from very slow when it's moving through the tiny capillaries, the blood are basically just forcing their way through the channels because they're actually bigger than the diameter of the channels, to when it's in your artery coming out your heart, it's moving incredibly fast. And it's likely that in the time it takes for invasion to occur, the parasite could have made it basically the whole way around your body. So it's likely to experience all those different flow rates. So if we could understand what flow rates are best for invasion, we might understand where in the body invasion might be occurring.
Okay, that's interesting. Yes, please.
Hi, I want to ask you a question, Itai. I was wondering with your molecule, which can change its diffusion speed with the different UV amount you apply to it, whether you can create one of the synthetic touring patterns. So one of the conditions on creating these patterns is different. If you have 2 different morphogens that are activating or inhibiting each other, but they also should have different diffusion speeds, whether you can create the system.
Okay, I think I need you to clarify the question. I mean, yes, I am changing the diffusion of the system with increasing UV exposure time, so it creates more cleavage, and then I can tune it. But what did you mean exactly?
Whether you can create a synthetic like a touring pattern.
What do you mean a toy pattern?
So tiger stripes, zebra stripes, they all kind of potentially arise from this networks of interacting signaling proteins which activate and inhibit each other.
Okay, I think I understand. So I'm not sure because this would be more of a kind of a crystal structure, I think. When it's liquid phase separation, as I said before, it's dynamic and it's diffusive with the environment. So that's why when I irradiate a single droplet, I can make the release of the cargo, and then the cargo, it remains intact, and the cargo can go back inside afterwards because it diffuses. Not only the cargo can diffuse, but also new peptide that was not cleaved can go back inside. So it's always diffusing. So to make patterns, it's I think it's kind of impossible, but I wouldn't say that.
Thank you.
Yeah, we have 1 more minute. Yes, please.
My question is to Emma. And during your presentation, you said that to study the effects of the environment on the rate of invasion, you started shaking the sample, basically. And I'm just curious, what is the environment you're trying to replicate? It's just, why shake it? Or is it next time my doctor asks me to just stay put, I shouldn't sneak out and start running, and some other things I might kill myself by increasing the rate of invasion?
The reason we chose that sort of motion was because in the literature it said that basically that increases the invasion rate. It's quite common when you're doing cell culture to shake to increase the oxygen diffusion or the contact between the cells. And it was particularly the contact between the cells I was interested in because I'd done all these assays with the tweezers where I was looking at the force of attachment, but basically when it happens in static, the parasites can just fall out of a schizon and bump into the surrounding cells, and there's nothing moving, there's no kind of challenges where you'd need more attachment force. So the idea was the shaking would increase the contact between the cells and make it harder for attachment to occur. And I've actually shown that when you disrupt some of the proteins that I showed were important to attachment, that impact on shaking is more significant, suggesting that the shaking is making it harder for those attachments to occur. But it's definitely not like a great mimic for the body, or the shaking is a very crude way of doing this, but it means you can test hundreds of conditions in parallel, which just wouldn't be possible with a microfluidics device.
But so you're saying that this method is actually quite common and people just shake their...
Yeah. I think a lot of the ways we culture cells are like they're not representing the biology really. It's just someone in a lab has realized that when you do this to your culture, something changes, and we can measure those effects, and then we can try and understand the physics of it. But when you try and understand the physics of what the liquid is actually doing, it's incredibly complex across the well it's changing. So it's a very crude way, but we see it changes the biology. So it's an interesting thing to look at. But the reason I tested all these different speeds was all papers I found just said shaking. They never defined the shaking speed very precisely. And so I wanted to understand whether different shaking speeds had an effect, to kind of tell people in the field that we need to be considering our conditions that we're culturing in more carefully, because they can impact the biology.
Okay, we need to finish. Thank you. Thank you for listening.